Hello, I'm Walt Storkus. I'm professor of dermatology, immunology, pathology, and bioengineering in the School of Medicine. I'm also a member of the UPMC Hillman Cancer Center. While I had early aspirations of becoming a mathematician, that quickly gave way and I became an immunologist and joined the UPMC faculty in 1991. I've received continuous NIH funding since 1994, and the lab is currently supported by R01 and PO1 awards. Over the past 25 years, we've developed 7 patents, published more than 250 papers. My major research interests lie in the areas of cancer immunology and the development of effective cancer immunotherapies. This interest mainly focuses on 3 cell types: dendritic cells, T cells, and tumors. With regard to how one can use dendritic cells best to elicit therapeutic T cell responses in patients, how to direct those activated T cells into the tumor microenvironment, and then lastly, how to protect and sustain their function so that they can mediate greatest therapeutic benefit in the tumor microenvironment. With regard to immunotherapies, we stressed the development of cancer vaccines, more recently vaccines targeting tumor-associated vascular antigens that are recognized by T cells over the past 5 to 10 years. In addition, we want to develop combinational approaches that integrate vaccines as well as other immunotherapeutic modalities, including checkpoint molecules, to achieve greatest therapeutic outcome. In recently funded studies, again, speaking to the vascular targeted vaccines, tumor blood vessels are very different from the blood vessels in normal tissues. They are impacted by the local tumor microenvironment, which is hypoxic, acidotic, and has high interstitial fluid pressure. These stressors promote an altered epigenetic programming in these cell types within the tumor microenvironment, and this leads to overexpression of antigens which are not normally found in blood vessel cell types. These proteins can be degraded into small peptide fragments that are presented in MHC molecules and can be recognized by killer T cells, which can mediate anti-tumor function by direct cytolysis as well as production of pro-inflammatory cytokines. And these T cells can be elicited through specific vaccination, or they can be expanded ex vivo from patient peripheral blood, and then these particular T cells can be adoptively transferred into the patient to mediate cure. Our preclinical modeling data was then translated into the development of a phase 2 pilot trial in which patient apheresis was used to generate monocytes. Monocytes were then differentiated into dendritic cells, which were loaded with a mixture of blood vessel antigen-derived peptides. And then this vaccine was injected intradermally into the patient every other week for several weeks, along with the adjuvant dasatinib. This vaccine was shown to be immunogenic in a subset of patients, here showing you specific T cell response to vaccine peptides, along with PET-CT imaging indicated partial responses at these time points, and the regression of lung metastases in this particular patient 1 on trial. Overall, the key findings in this protocol were that we achieved a 31% overall response rate in checkpoint refractory melanoma patients. Tumor regression only occurred if the patient developed specific T cell response to vaccination, and the regressing tumors had evidence of robust immune cell infiltration. Another unexpected finding in this particular study was that females responded far better than males, approximately 4-fold better than males. And this was also age dependent in the females, so that younger females tended to exhibit much longer progression-free survival than older females. This data was recapitulated in animal modeling data, and it suggests the dominant modulatory action of sex steroids, which we're currently investigating. And then lastly, in an R01 and PO1 that are ongoing, we are investigating immunotherapies predicated on injection or treatment of tumors with IRF3 agonists in order to promote homeostatic chemokine release. It's expected that this chemokine production in the tumor will recruit T cells, B cells, and dendritic cells from the peripheral blood, and that if appropriately robust and sustained in the tumor microenvironment, this can lead to the development of so-called tertiary lymphoid structures, These are large, non-encapsulated lymphoid-like organs that largely mimic lymph nodes in structure. They have B-cell regions as well as T-cell regions that are important for the development of anti-tumor antibody responses, as well as the differentiation of anti-tumor cytotoxic T-cells, which can then go on to mediate regression of tumors. With the overall project goals being to effectively turn tumors into lymphoid organs for therapeutic benefit, and then to develop novel improved immunotherapy protocols for patients with advanced stage disease that are typically resistant to standard of care checkpoint blockade. We've developed and patented vascular vaccines that are capable of targeting any form of solid vascularized cancer and provided proof of principle data for safety and bioefficacy of these particular strategies in melanoma patients. We've developed the novel gender dimorphism data in melanoma patients that speaks to sex steroid modulation of immune responses that may need to be counteracted or amplified in the case of females to accentuate the benefits of these approaches. And then lastly, we've developed treatment regimens that are capable of converting tumors into inflamed lymphoid organs in association with therapeutic benefit. Future directions include development of novel vascular vaccines based on novel single-cell RNA-seq data coming from vascular endothelial cells and pericytes from the most common forms of cancer to better dissect the mechanisms underlying genetic dimorphic responses to vaccination and the impact of androgen receptor blockade or estrogen supplementation. And then lastly, to really determine how tertiary lymphoid structures impact the local immune cell fate, function, and specificity, and how this relates to clinical outcomes in next-gen clinical protocols. Thank you for your attention, and please feel free to contact me at this email address should you have any questions. Thank you.